All right. Good afternoon. I'm Edward Guo, professor and chair of the biomedical department at Columbia University. Today, we are very excited to continue our research spotlight series, Faculty BME Faculty in Focus, where we will highlight the exciting research conducted by our own Columbia BME faculty. These lectures are all available on our BME YouTube channel at youtube.com/Columbia BME. Throughout the presentation, if you have a question for our speaker, please feel free to type them into the Q&A area at the bottom of your screen. We'll try to answer as many as possible at the end of the lecture. At this time, we would like to introducing our BME faculty in focus speaker today, Assistant Professor José McFaline-Figueroa. José's research focuses on defining the molecular changes induced in cancer cells after exposure to anti-cancer therapy, how those changes alter response to the treatment, and how they differ as function of the genetic background of individual cancer cells. The goal of his laboratory is to leverage this functional atlas of cell response to arrive at a novel treatment against cancer, with particular focus on aggressive tumor types that frequently fail in the current standard of care. José's approach, including the development of application of tools centered around single-cell genomics, multiplex genome editing, and chemical genetics. These techniques increase the scale at which we can determine how chemical and genetic perturbations alter molecular phenotypes and how those phenotypes vary due to the cellular heterogeneity observed between and within tumors. José received his bachelor degrees in chemistry from the University of Puerto Rico at the Mayagüez, and in 2006, and his PhD in biology from MIT, and in 2014, he was a postdoc fellow at the University of Washington before joining the Columbia I think in 2020, end of 2020 or early 2021. So welcome, José.
Thank you. So thank you so much, Ed, for the kind introduction. Yes, so early 2021, we started the lab here at Columbia BME. And so today, let me start sharing slides. So first of all, it is an absolute pleasure to be able to present some of our work to all of you. So for today, what I wanted to do is give you kind of an overview and some examples of some of the approaches that we've developed in the past to define cellular response to exposure with a particular focus on cancer. And then towards the end, I'll highlight some of the areas that my lab is working on now. So broadly, our interests lie on a couple of areas, but very heavily focused on cancer research. This is a figure from the Hallmarks of Cancer from Hannah Weinberg. And when we look across these types of annotated lists of cancer processes, we are interested in determining what are the genetic determinants and pathway requirements of these cancer phenotypes, really with the goal of using that information to inform therapeutic strategies in the disease. So that is at a very 30,000-foot view, we want to understand how are cancer-associated processes regulated? Can we use this knowledge to inform successful interventions? And if we kind of understand these processes, or essentially understand how they occur, we can use that information to inform therapeutic strategies for the disease. Now, I'm also interested in defining the response of cancer cells to already established therapies. So currently we have a pretty impressive arsenal of compounds that we can use to treat cancer. And these compounds elicit a really wide variety of different responses, and they depend on many things. Of course, they're going to depend on the mechanism of action of the drug and the particular target, but they can also vary as a function of genetic background, both between tumors and within an individual tumor. And you can imagine that this heterogeneity can manifest itself, and it frequently does, as subpopulations of cells that are refractory to treatment and can give rise to tumor recurrence. Now, overall, this is a very simplified view of cancer-associated processes and how to target them. So in reality, if we zoom into any of these phenotypes, there are a very large number of different genes and pathways that mediate these responses, which means that there are a large number of possible therapeutic options, and therefore each might be associated with different mechanisms of resistance. So really, our goal has been to develop and apply tools that really allow us to increase issues with complexity and scale that arise from this interaction, to be able to ask other questions, including what defines a response of cancer cells to therapy. Again, how does it depend on genetic backgrounds, and can we gain insight into what are putative mechanisms of resistance? So the way that I like to think about the lab is really, what we're trying to do is leverage single cell technologies. These are technologies that allow us to treat every tumor cell as a distinct entity, to define the regulation of cancer-associated processes and the response to therapeutic intervention. So today I'll talk about past work using single-cell genetic screens to define how genes regulate a cancer-associated process. So that is, when we're looking at a population of cells, can we genetically perturb different parts of a particular pathway, and ask how does each one of those genes contribute to a complex phenotype, in this case, gene expression. And gene expression with regards to some biological process. And in the second half, I'll talk about our development of a platform to define how cells respond to drug exposure in a manner that accounts for heterogeneity across that response. So I'll begin here with giving you a brief story of using single-cell genetics to define one biological process, and then we'll switch over to chemical transcriptomics. So in particular, so this is a prior study in which we use single-cell genetics to identify the regulation of the epithelial to mesenchymal transition. Now, the epithelial to mesenchymal transition, or EMT, is the transition by which cells transition between two somewhat extreme cell states. So on one end, we have an epithelial cell, which can be found in highly organized sheets. It displays apical basal polarity. It has very strong cell-cell interactions and generally has low motility. On the right, we have a mesenchymal cell, which displays front-rear polarity. It has little to no cell-cell contacts. It is very highly migratory, and it's capable of invading tissues. Now, in between these two states, it's very well appreciated that there is such a thing as an intermediate EMT phenotype. Now, even today, it's still somewhat debated what the nature of that intermediate EMT phenotype is, and how we can best describe it. And fairly simple questions, including, is intermediate EMT a discrete step in a process? Is there a continuum of phenotypes that go from the epithelial to the mesenchymal side? Now, EMT overall is very important for proper organismal development, wound healing, as well as pathogenesis. So it is associated with different disorders, including the induction or the aberrant induction of fibrosis, which itself has been shown to involve a partial EMT phenotype, and has been implicated, of course, in the dissemination of cancer cells during metastasis, so mesenchymal cells and intermediate EMT cells being able to invade tissues to colonize other sites. So really understanding the nature of this process can help us understand human organismal development more generally, and human disease. Now, to define the nature and the regulation of an EMT, I developed an in vitro model, a spontaneous, spatially determined EMT. And to generate this model, we took advantage of the fact that some epithelial cell lines, if they are cultured at low confluence, or that is if you remove or have low cell-cell contacts, some will spontaneously undergo an EMT. So in this particular assay, we start the assay by seeding epithelial cells within cloning rings as a very high confluence patch in the center of a culture dish. Then we remove the ring and we allow the cells at the periphery to sense that empty adjacent space and undergo a spontaneous EMT. So essentially what we're looking for is to create a colony where at the center, you have cells that have very strong epithelial phenotype, and at the edges, they have mesenchymal traits. So here's a bright field image of a colony from this assay. So this colony has been fixed, stained with crystal violet, which is the dye that just generally binds DNA. So in areas where you have really dark staining, these are areas where you have a lot of DNA, a lot of cells packed together very tightly, so that epithelial sheet, and you can see that it decreases as you go out from the center. Now, if you use immunofluorescence, we can look specifically at the expression of a marker of epithelial cells, in this case, E-cadherin. And we see that E-cadherin staining, which is here in green, is very high in the center of the colony. And then it decreases as you move away from the colony. So it does seem that we are getting that spontaneous EMT phenotype. Now, we next dissected these colonies into two fractions. So we wanted to have some ground truth about the fact that the cells in one area are more likely or should be more likely to be epithelial, whereas on the other, they should be more mesenchymal. We took each one of these fractions and we subjected them to single-cell RNA-seq analysis. And really, single-cell RNA-seq analysis here is really crucial to avoid averaging over these heterogeneous populations. Because these assays are essentially able to treat every cell as a distinct entity. Now, in addition, we do expect to get a continuous progression along an EMT, at least as a function of the radius from the center of that colony to the very edge. Therefore, we subjected single-cell transcriptomes to single-cell trajectory reconstruction. And the goal here is to be able to recreate the trajectory of cells going out from the center of the colony, which in turn, we hope is telling us something about EMT progression. I'll show you that, in fact, in this case, it does. Now, this is the resulting trajectory. So this particular trajectory was built using reverse graph embedding as implemented in Monocle 2. So in this particular trajectory that we recovered, we obtain a largely linear trajectory where there's an enrichment of cells from the inside of the colony, from that inside fraction on one end, and enrichment from cells from the outside of the colony on the other end. Now, we can define a measure of progression along this type of trajectory. In this case, we're calling it a pseudospatial trajectory that can tell us about what processes are occurring as you go across this trajectory. And we wanted to ask whether epithelial and mesenchymal markers varied in the expected manner. So here I'm showing you expression of the epithelial marker E-cadherin as a function of pseudospace, and we see that it decreases as pseudospace progresses, which is consistent. CDH1 again encodes for E-cadherin, the protein that we saw in the immunofluorescence. So we're getting a similar answer that as you move away from the center of the colony, you have lower E-cadherin expression. And conversely, if we look at a marker of mesenchymal cells, vimentin, we see that it sharply increases as you go from the inside of the colony to the outside. So we are indeed essentially creating an EMT trajectory when we do this process. Now, we performed many experiments, one to show that that was indeed a continuous trajectory. And in addition, that multiple additional experiments that we did in other lines really seem to highlight the fact that EMT is in fact a continuum of cell states. So that is that cells continuously vary as they go from a very epithelial characteristics to very mesenchymal. And what's nice about this is that you're identifying these states as a function of the entire transcriptome, not just select markers. Now, we used pathway analysis to identify what pathways are differentially expressed as cells progress along this trajectory. And I'm going to highlight a subset of those results. So one we observed that two waves across the trajectory, so two modules of gene expression that are associated with RAS signaling that occur as a function of EMT progression, one as cells exit an epithelial state, and a second where they acquire mature mesenchymal phenotype. So these are two gene expression modules that are distinct from each other, but both have characteristics that in that they're both characterized in the fact that they have been associated with active RAS signaling. Now, when we think about this result, as well as all of the pathways that we see that are dynamic as a function of EMT, we started to think about, well, can we reconcile some of the observations about EMT being discrete or continuous? Specifically, our model kind of implies that particular signals, or results imply that particular signals mediate progression along the EMT continuum. And what that might mean is that if you lack a particular signal, then this will lead to accumulation somewhere along the EMT trajectory, which will give you something that looks like a discrete cell state. Now, interestingly, this was consistent with reports at the time that identified diverse partial EMT phenotypes in human head and neck squamous cell carcinoma. So the hypothesis there is that those different partial EMT phenotypes are due to tumors lacking different signals along EMT progression. So we wanted to test whether we could stop cells at different areas across an EMT by removing some of these signals. And to that, I want to transition to introduce single-cell genetic screens. And these single-cell genetic screens via CRISPR-Cas9 really take advantage of what I say this a lot. This is one of my favorite characteristics of single-cell genomics experiments, and that is that you can treat cells as a container for experiment. So in this particular assay, you begin with Cas9 expressing epithelial cells. You can deliver guide RNAs targeting a gene of interest. And then after sufficient time for gene editing, you can ask, well, how does genetic perturbation alter a phenotype of interest? For example, progression along an EMT. Now, the fact that this is a single cell screen means that we can do this for multiple different genes at the same time and ask how many genes contribute to the phenotype in parallel. And this is really thanks to landmark efforts developed by multiple labs now 5 or 6 years ago that allow us to identify the nature of a guide RNA within the context of a single cell CRISPR screen. And in the past, we have optimized some of these methods to perform different screens. For example, so I won't talk about it today, but we've looked at the molecular consequence of tumor suppressor loss on the responsive cells to DNA damage. So for this screen, we focused specifically on cell surface receptors and transcription factors that are involved with an EMT. So, this isn't a screen for novel regulators of an EMT. This is a screen to determine where along the EMT continuum is the activity of a known EMT regulator necessary. So can we place them along that trajectory? So I want to give you a little bit of intuition as to what it is that we're looking for. So let's think of the case where you have cells that are expressing control non-targeting guide RNA. So these cells should be able to transition fully across the continuum in whatever manner and whatever rate is quote unquote normal for these cells. And what we're looking for are knockouts or gene editing events that lead to an accumulation across this trajectory. This could be a very strong block to progression, or it could be something more subtle, like a change in rate. So that is loss of that particular gene does not abrogate progression along an EMT, but perhaps if there's redundancy in a particular area, loss of one signal will change the speed at which cells move across this trajectory. Now, I'll start presenting by presenting control that we built into the experiment. So in addition to our spontaneous EMT model, we also expose cells to TGF-beta, which is a potent inducer of an EMT. And TGF-beta binds TGF-beta receptors, activates SMAD signaling, and leads to the block of epithelial traits and induction of mesenchymal traits. So therefore, we would expect that in control cells expressing control guide RNAs, TGF-beta should push them far into the trajectory, whereas knockout of TGF-beta receptors should really be a strong block to TGF-beta induced EMT progression. So here I'm showing you now data. This is indeed what we observed. So we have the density of cells along EMT progression. Again, this is TGF-beta driven EMT. We see that control cells make it far into the trajectory, and cells that are genetically perturbed for TGF-beta receptors accumulate very strongly at the beginning of the trajectory. Now, I will note that you see this subset of cells that do make it quite far into the trajectory, and this likely refers populations of cells where we did not achieve complete loss of TGF-beta receptors. And this is something to keep in mind with this type of screen. So identification of a guide RNA within a cell does not equate successful loss of function of that particular gene. So when we're doing gene editing, we do different approaches to be able to account for this mix of true loss of function and wild type cells. Now, going back to our spontaneous EMT, here we have again the distribution of control guide RNAs, but now across that spontaneous trajectory. And I'm focusing in on this one region where we see much more subtle accumulation of cells that are associated with loss of an EGFR, of the EGFR receptor. Now, this is an area where we see more of a subtle change in rates. Because it was more subtle, we wanted to validate it. So we treated cells with an EGFR inhibitor and then measured by flow cytometry levels of epithelial markers and mesenchymal markers. And you see that an EGFR inhibitor in this cell line can block spontaneous EMT as measured by these markers, and including blocking increase in vimentin. And this is just some of the flow cytometry plots for these experiments. Now, EGFR was one of 3 signals that we identified in that area that were associated with that type of subtle accumulation. We also identified MET and FGFR, 2 other receptor tyrosine kinases whose loss led to the kind of subtle phenotype. Now, all of these kinases are upstream regulators of RAS GTPase. And really what this kind of highlighted to is that this RAS-MEK axis is really integral to multiple parts of an EMT, specifically exit from that epithelial state and induction of that more mature mesenchymal state. Now, we did test this. So we looked at the accumulation of cells along an EMT as a function of MEK inhibition, and they do accumulate at the beginning of the trajectory in an epithelial state. But we also asked, well, what happens when you start playing around with the different signals that a cell receives? Can we purposefully make partial EMT phenotypes? So as one example, we treated cells with both a MEK inhibitor and TGF-beta. And here, what we observed was accumulation of cells in an intermediate EMT phenotype. So the MEK inhibition is blocking cells from going towards the later, so that second wave of RAS signaling is not occurring, but TGF-beta is able to help go through that first gate. So this is an example where distinct combinations of signals can give you distinct phenotypes across biological trajectory. Now, we also wanted to ask, well, is there evidence for this type of RAS-MEK checkpoint in vivo? So we looked to recent single-cell RNA-seq data set from Aviv's Regev's lab. So this is the partial EMT phenotypes in head and neck squamous cell carcinoma that I alluded to earlier in the talk. And they found that these tumors express diverse partial phenotypes. And we wanted to ask, well, do we see any of the gene expression changes associated with these waves across these tumors? So first, we took genes from these associated with each one of these 2 modules. So here I'm showing you data for the second late RAS-MEK checkpoint. So first, if we score our cells, in our trajectories for this group of genes, we see what we expect, where we get an increase as you go from the inside to the outside of the colony, so earlier late pseudospace. And TGF-beta, that potent inducer of an EMT, can increase the level of these both in early and late parts of the trajectory. Now, for the tumor samples, here they are ranked according to the partial EMT phenotype observed in that initial study. And we do observe a nice positive correlation between a tumor that has lower partial EMT rank and having the lowest score for this late wave of RAS-MEK signaling. And we observed pretty much the same pattern with that earlier wave as well. So this does make an argument for perhaps using MEK as a therapeutic strategy to minimize EMT-associated cancer cell dissemination. But I'll note that targeting MEK and other kinases can frequently resistance is observed in the clinic. So in the lab, we are also interested in taking and understanding what drives resistance to these types of therapies. So essentially, can we also go one step forward and think about what are likely mechanisms of resistance? So for this part of the talk, I showed that we developed an in vitro spatially determined model of an EMT, and that EMT is continuous in nature. We use a genetic screen to identify signals whose loss impedes EMT progression, and that MEK signaling mediates progression along multiple points in an EMT. And I'll leave you, for this part of the story, I'll leave you with one more thought, and that is the fact that, we're observing that lack of signals can give the appearance of discrete stages. So for me, at least, it starts to ask questions about cellular ontology. When we're thinking about cell types or cell states, how do we define those in the context that we might be seeing pieces of continuous processes? So now we'll switch over to our work on multiplex chemical transcriptomics, and a platform to essentially perform high throughput screens at single cell resolution. Now, I'll start by introducing high throughput screens. So there really are a cornerstone of the pharmaceutical drug discovery pipeline. They enable the characterization of biochemical efficacy, potency, cytotoxicity of many bioactive compounds. Now, these screens usually occur in plate format. I'll give you kind of one example. So let's say we have a 96-well plate with 12 compounds at 8 doses, and given some readouts, we can recover a dose-response relationship. Now, the power in these screens is that you can do this across a large number of plates, say across different genetic backgrounds, and then ask how does genetic background shift those dose-response relationships. Now, many of these screens have been coupled to what we can think about as gross phenotypic or very, very specific molecular readouts, so things like viability and proliferation, or reporter expression, maybe enzymatic activity. And you can imagine that this does limit what we can learn about things like mechanism of action and off-target effects, although they are very, very powerful. Now, more recently, there have been efforts, including here at Columbia, to couple these high throughput screens to a bulk expression analysis. And by bulk, I mean you take all of the cells that are treated with a given compound, and then you generate a gene expression profile for that one sample. So this is giving us a pretty wide molecular view of what occurs upon perturbation, but this is not as amenable to the study of complex population. And in addition, if you have instances where drug exposure leads to more than one response, that is when you see heterogeneity in response to exposure, that readout that you're getting is essentially an average of whatever cell states are being induced. Now, this is a talk about introducing single cell technologies. So yes, I'm trying to convince you that single cell transcriptomics can overcome some of these issues that are associated with the analyses of this type of complex population. And there are some methods that can scale to the magnitude necessary to perform a high throughput screen. Now, single cell combinatorial indexing, scRNA-seq, developed by Junye Cao and Malti Spielman in the Shunduri lab. Junye Cao now has a lab at Rockefeller University close by, has incredible throughput. and it allows for the analysis of millions of cellular transcriptomes. And recently has been used to profile many cells across different conditions. For example, 2 million cells during mouse embryonic development. More recently, it has been used to develop both gene expression, increment, and accessibility maps of human fetal development. So it really is quite a powerful approach to profile large numbers of cells, so in the millions. However, it's not straightforward to parse that throughput across a large number of different conditions. So we therefore sought to create a method that essentially did that could couple a high throughput screen to high throughput single cell RNA-seq. Our approach is similar to how you would start most of these screens. So we begin with cells in well format, exposed to many different perturbations. Then we lyse cells to expose nuclei. And we did this because of an observation that we had that nuclei have, just fantastic affinity for single-stranded DNA. Probably others have observed this as well. But this gives us an opportunity to affix barcodes to nuclei. And these barcodes are oligonucleotides that essentially have all of the goodies that a cell needs, all of the goodies that we need to be able to capture that oligonucleotide within the context of a single cell RNA-seq experiment. Now, in addition, it also has a barcode that relates back for every cell, which well, and therefore which condition a cell was exposed to. So this then allows us to pool all of our conditions together, whether it be thousands of unique treatments, and perform common spiral indexing RNA-seq. And I'll note that this type of method is similar to other cellular hashing strategies. However, our approach necessitates only oligonucleotides, so it's fairly inexpensive, which is what you want if you want to scale up a lot. And it is coupled with a method that can be used for high throughput screening. So first, to ensure that we could use this barcoding approach to determine the effect of various compounds on cellular transcriptomes, and within one experiment, we treated A549 lung adenocarcinoma cells to 4 small molecules of target regulators of transcription: an NF-kappa B inhibitor, a corticoid receptor agonist, a p53 agonist, and an HDAC inhibitor. So here I'm presenting to you a UMAP embedding for this experiment. So for those that aren't familiar with UMAP visualization, UMAP is looking to summarize or visualize, say, the relative relationships between cells in lower dimensions, so as opposed to high gene expression space. So here, every dot is a cell, and the cells are colored by the treatment to which they were exposed. So we do see from this view very clear segregation of cells that were treated with different compounds. We can also observe dose dependence in the association or the distribution of UMAP space. So if we color cells by dose, we see that as we increase the dose, the cells start taking up different parts of the UMAP. And we can keep going with higher doses. Now, one thing that I'll point out as well is that the way that we designed this experiment, we try our best to maintain the relative proportion of cells within the experiments. And what this does is that it allows us to recover viability dose response estimates. So, within the context of this large scale molecular screen, we can recover some information associated with more classical high throughput screens for proliferation or viability. So here I'm showing you 2 examples of inferred cell counts to 2 agents, an NF-kappa B inhibitor and a p53 agonist. Now, of course, what we wanted really to do was to demonstrate that our approach could scale to a magnitude that's on the order of a chemical genetic screen. So we devised a screen where we took 3 cancer cell lines: A549 lung adenocarcinoma, Ki562 myelogenous leukemia, and MCF7 ductal carcinoma, and we exposed these cells to 188 compounds, targeting a really wide variety of different mechanisms of action. So about half of our library was devoted to compounds that targeted the transcriptional machinery. So we know, or we expect, that we should get a a change in gene expression that tells us something about mechanism of action, or that's closer to its mechanism of action. But we also, the other half was really devoted to many different types of compounds. And here we wanted to ask, well, can we learn something about mechanism of action for things that are quite far from the transcriptional machinery itself, or at least not directly modulate transcription. And we did this across 4 doses and multiple replicates for, in this particular screen, we had around 4,600 unique conditions. There were actually a couple of extra ones in there, so closer to 5,000 in this particular experiment. And in all, we collected a little bit less than 600,000 cells. And again, this is all within the context of one experiment. So first, I wanted to demonstrate that we indeed are able to recover information about mechanism of action from single-cell chemical transcriptomics. I think this is a lot more standard today, but still we wanted to show it at the time. So here are 3 clustergrams of essentially trying to summarizing the similarity of cells exposed to different compounds. So I'm going to focus in on one of these, the A549 line. So if we first focus in on this clade is composed of cells exposed to HDAC inhibitors. So again, this makes sense that we can get a kind of a similar readout for something that directly alters transcriptional processes. But we also see, again, clades made up of compounds that do not directly target transcription. This is a clade of our kinase inhibitors. This is a clade of receptor tyrosine kinase and MEK inhibitors. This is a clade made up of HSP90 inhibitors. So we are able to, from this type of, single-cell RNA-seq data is sparser than bulk expression, but we can recover these commonalities quite well. So I'll switch over now to highlighting some vignettes across our experiment, and essentially highlight where single-cell resolution is really key. So things that we could only identify with single-cell RNA-seq. So I'll start with giving you an example of when we observe heterogeneity in the response within a cell line and within one drug dose combination. An instance where we see differences between cell lines, and this has to do with the genetic background of the cells. And an instance in which we do see heterogeneity in response, similarity across the cell lines, across many compounds, and it gives us insight as to how cells respond to a class of compounds. So first, I'll talk about our observation of where cells respond heterogeneously to a drug dose combination. Here I am presenting UMAPs of the A549 cell line exposed to microtubule stabilizing agents, apophyllin A and apophyllin B. And in response to these drugs, we observed that at pretty much all doses, we had multiple pronounced types or essentially groups of gene expression changes across cells in one drug dose combination. We can visualize these patterns using UMAP. So here we have 3 boxes where cells that are treated with these drugs accumulate, and each one with their separate kind of transcriptional consequence to treatment. Now, these agents target microtubules, were associated with multiple cellular processes, but are really key in mitosis for the proper separation of chromosomes. So we first wanted to ask, is there an association between cell cycle effects and response to these drugs? So if we look at those 4 blocks, and now the same UMAP is colored by the expression of genes associated with proliferation or proliferation index, we see that 2 of these boxes are in areas of high proliferation, 1 in an area of low proliferation. So we do see an association there. And actually, if we go a little bit farther, one of these boxes is associated with being in the G1S phase of the cell cycle versus G2M. So we do see heterogeneity in response depending on where in the cell cycle a cell is. Now, going back to the larger data set, here I'm showing you now 3-dimensional UMAPs of this large screen for K562, A549, and MCF7 cells. For each, we have vehicle-treated cells in gray, and then we have treated cells in red for K562, blue for A549, and yellow for MCF7. Now, I'll begin, I'll show you as well, so in this particular view, depending on the particular treatment, cells segregate to different areas of the UMAP, and some do so depending on the genetic background of the cell. So here I'm showing you the same embeddings, but now if a cell was treated with a MEK inhibitor trametinib and has a significant change in gene expression, we colored that cell in this light blue, and everything else is in gray. So from this view, you'll notice that there's a blue cluster completely missing from MCF7. So MCF7s do not respond particularly strongly to trametinib exposure. And this is likely a consequence of the genetic backgrounds of these cells. So K562 cells have a driver oncogene that is BCR-able, a tyrosine kinase fusion that renders it constitutively active. A549 has activation of KRAS, and MCF7 has an activated version of PI3K, the catalytic subunit. And these different driver oncogenes turn on different pro-survival pathways in the cell. So BCR-able is able to turn on both the MEK and AKT pro-survival pathways. KRAS is more strongly associated with MEK, and PI3K with the AKT pathway. So it makes sense that in the first two cell lines, abrogation of MEK activity leads to a stronger transcriptional response. Now, the last vignette that I'll go through has to do with the response of cells to HDAC inhibition. So here I'm showing you those same UMAPs, but now cells colored by the type of compound that they were exposed to. We have HDAC inhibitors in green, and we have these projections of cells that were exposed to HDAC inhibitors, to one of 18 different HDAC inhibitors, the tips of which were enriched for cells treated with the highest doses. So we hypothesized that there was a drug-induced trajectory being formed, and we use a mutual nearest neighbor approach to collapse all three cell lines that are vehicle or HDAC inhibitor exposed into one common space, and that's this joint trajectory that you see here. So in this joint trajectory, vehicle exposed cells are on the left, and as cells respond to HDAC inhibition, they move towards the right. So, whoops, clicked the wrong thing, towards the right. And we can see that as we, I'll give you one example of that occurring. So here we have cells exposed to the HDAC inhibitor persinostat at a low dose, and they're mostly on the left, to your screen, to the left side of the trajectory. And as they respond to higher levels of the HDAC inhibitor, they move towards the right. And you'll see that it's pretty clear to see here where the trajectory is coming from, that continuous progression. So you see that some cells are still at the beginning of the trajectory, and some have made it quite far with this dose of drug. And as we increase the dose, we can push cells farther and farther in. Now, this trajectory is associated with an exit from the cell cycle, as well as, oh, sorry, I wanted to make sure no one was telling me something about the webinar. Exit of the cell cycle, as well as an increase in the expression of genes associated with metabolism. Now, this increase in metabolism genes really pointed to a particular type of metabolism that seems to be perturbed as a function of HDAC inhibition. Now, histone acetylation is more frequently associated with the regulation of gene expression, where acetylated histones are a marker of open chromatin, which is more transcriptionally active. And HDACs really fulfill that role of shuttling cells towards the closed chromatin state, where that now you've abrogated or you brought back that positive charge in the histones, and you get really strong association between the DNA and histone proteins, and you get lower transcription. Now, a less appreciated fact is that histones also serve as reservoirs for cellular acetate. Now, acetyl-CoA, which is derived from acetate, the product of HDAC class 1, 2, and 4 histone deacetylation, has important roles in metabolic homeostasis, and inhibition really prevents recycling of chromatin-bound acetyl groups for many processes. Now, this is a pathway diagram of many different processes in the cell that will increase, or enzymes that will increase acetyl-CoA pools, and we observed increased expression of many of these as a function of treatment. Now, first of all, we did confirm that at the doses that we were looking at, we were getting robust accumulation of acetylysine across the cells, which is what we expect. But when we wanted to ask the question, well, if this is associated with acetate metabolism, then if we supplement with acetate within the media and treat with an HDAC inhibitor, then we should be able to rescue part of that molecular progression across the HDAC inhibitor trajectory. So here's one example. We did this experiment across our other cell lines as well. So this is cells treated with a high dose of the inhibitor and increasing levels of acetate. And we can see this robust accumulation of cells at the end of the trajectory when they're not treated with acetate, and we can block some progression as we increase the amount of acetate supplementation. We can also do this with pyruvate. We can also do this with citrate. And really what it kind of tells us is that at least a proportion of the sensitivity to HDAC inhibition is due to acetate deprivation, where HDAC inhibition leads to or pushes us to the cells towards an acetate limited state and cell death. And this can be rescued with acetate pyruvate. So to summarize this part of the talk, we developed sci-Plex, an inexpensive sample multiplexing strategy that's compatible with single-cell RNA-seq. And we applied it to perform high-throughput chemical transcriptomics of single-cell resolution, identifying heterogeneity in response to different cell types, and that the resolution helped reveal heterogeneity in response to microtubule targeting, HDAC inhibitors, and a couple of other examples that we found as well. Now, so I hope it convinces you of what are the advantages of using this type of method. So in my lab, we're using this type of platform to do many different types of experiments. So we can use this to determine how many different heterogeneous models respond to many different agents. So I do want to leave you with an idea of the type of questions that my lab is now investigating in the context of defining the molecular regulation of cancer-associated processes. Now, we are continuing with our large-scale chemical profiling to define the response of cancer to therapy, focusing on as well on determining the nature of how the nature of a compound, I should say, against a single genetic alteration affects tumor molecular responses and resistance to therapy. So not only taking large library of compounds, but even for one particular target, what is the world of different responses to targeting that via different modalities? We are also combining single-cell genetic screens with our chemical genomics approach to be able to define how individual genes contribute to the molecular response to anti-cancer therapies, both in vitro and in the complex context of the tissue. And the last area to highlight is the profiling of distinct cell-cell interactions in order to better understand how, take a reductionist approach to understand how interactions between cells leads to differences in response to therapy, and how those responses themselves in combination with cell-cell interactions depend on the genetic architecture of a tumor cell. And overall, we aim to be synergistic with other efforts to identify novel treatments in cancer. Now, we aim to apply these techniques to dissect the response to exposure in neurological disease, specifically in the context of neurodegeneration and neurological malignancy. For example, Ross Giglio in the lab has been investigating how genetically diverse models of glioblastoma brain cancer respond to comprehensive panels of compounds targeting a frequently activated oncogene in the disease, while Anna Skonen in the lab is investigating the contribution of many genes to the dynamic changes induced by treatment with standard of care chemotherapy. In this case of neurological malignancy, that standard of care is temozolomide, an oral alkylating agent. So very soon, we'll be talking about these projects and other projects in the lab in greater detail. So with that, I'd like to give thanks and acknowledge all of the wonderful lab members in the lab. I am lucky to have a phenomenal group of trainees working on different aspects of method development and asking diverse questions about neurological malignancy and neurodegeneration. I would also like to, of course, thank my home at Columbia BME. I would like to thank people over at the University of Washington Genome Sciences, where I did my postdoc, in particular, Cole Trapnell and Jason Jury and their labs. as well as Sanjay Srivatsan and Vijay Ramani, who are co-authors for the sci-Plex study. I'd like to thank past sources of funding and, of course, current funding. So with that, I'm happy to take any questions that you may have.
Thanks very much, Jose. That's really fantastic. So we have some time for questions, and then one question from anonymous attendees asking about how the 3 UMAD maps are generated, or they're generated independently or generated together. They can do a quantitative comparisons.
Yeah, so these are not aligned. The 3D UMAPs were generated individually for each of them, each of the 3 lines. In the case of the mutual nearest neighbor alignment, we subsetted vehicle and ASEC inhibitor exposed cells, and then use that cell type label to be able to align using mutual nearest neighbors. But yeah.
Okay. Well, so for those audience, watching this one live, feel free to use the Q&A features to asking questions. So the one question I have now is how easy your research is applying to in a clinical settings, and for example, really getting the, I guess, difficult getting the samples from the patients. But how you can get in those sample for patients, apply those technology really to improve treatment, right? Because you can only get in the samples after, unfortunately, those patients dies, and then means doesn't work, and then with those one working is difficult to do, right? So how you play out in the clinical?
So I think there are different. I think there are different areas that can be synergistic with each other. So I think we're, a lot of the assays that we have, you have the advantage to be able to profile a large number of models and hopefully a substantial amount of genetic diversity across tumor types, and try to learn some of the rules about how cells respond. And perhaps then doing some kind of comparative analysis with a sample that comes from the patient would be the way to go. So essentially align or embed a patient's profiles to a much larger reference of response to treatment. That's one way that we could go about it. The other way we do have, our in vivo efforts, of course, are, focusing on mirroring models of cancer, but we expect that those can also start to give us more hints about the more complex interplays that occur in the context of a tumor. And there are other labs that have models that are, perhaps not the same type of throughput that we do, but are taking patient samples and directly querying them for a couple of handful of drugs. So you can imagine, I think that the labs of Peter Knoll, Peter Sims, and Jeffrey Bruce have been doing this for a while. And it's you can imagine that this might be kind of a pipeline where you do, perhaps you prioritize for a given sample against a reference database broadly where that tumor type should go, and then grab a small subset of different compounds for this kind of direct tumor response from a given patient. So essentially it could be part of a broader pipeline, I think, would be the way that I envision those particular assays being used.
Great. Without the further question, thanks, Jose, for doing the faculty in focus, and for the audience, please check out our website, our social media, and then we'll plan every month have a new speakers from our BME faculty presenting the exciting recent work. Thanks very much, and good evening.
Yep, thank you.